Our next speaker is Jeff Good.
All right, thank you very much for the invitation. This has just been an outstanding meeting. I want to tell you today about some relatively newer direction in my lab that we're working on genomic imprinting and speciation. So my lab, and I think one of the central questions in evolutionary genetics related to speciation, is to really get at this question of whether or not recurrent forms of intrinsic reproductive isolation, so things that go wrong in hybrids, reflect common proximate and ultimate mechanisms. And I think as a community, we've done a pretty good job of answering aspects of this question in hybrid male sterility, and there's been a lot of work on that, in particular showing that particular developmental processes in male reproduction tend to be recurrently disrupted in hybrids. But we actually know much less about this question in the context of hybrid inviability. And so we've been interested in jumping into this in mammals. And in mammals, if we're going to think about the first part of this question, the question of whether or not there are recurrent phenotypes that we could actually need to explain with a common genetic or evolutionary basis, abnormal growth comes to mind. So when mammal species are crossed, often there are growth effects, and so often the hybrid offspring are of unusual size, either larger or smaller than the hybrid offspring or than their parents. And in a subset of cases, so we want to know if this generally has a common genetic basis, and over again we see a particular kind of growth effect, and that is parent of origin growth effects. And so reciprocal crosses between species kind of famously demonstrated by this lion-tiger cross produces either a really massive offspring, this is the liger, which is a real thing, and then the reciprocal cross, so ligers are massive and probably terrifying, but tigons are actually pretty small, and they're smaller than the smaller of the two species, a lion. And so this particular recurrent pattern in reciprocal crosses implies a genetic mechanism. And so, in particular, it implicates placental development and function. So this placenta is this really fascinating organ involved in nutrient transfer, and so it really mediates exchange between the mother and her embryos. And disrupted placental function again and again has been shown to be associated with growth effects. And so there's this really good functional data linking disruption of placental function to these kinds of phenotypes. And then from an evolutionary perspective, this is the most rapidly evolving tissue in mammals. So it's really fascinating. And we think this is because it really mediates this inherent conflict between mother and offspring and between mother and father in the case of multiple paternity. So parental conflict should be really intense here. And probably not coincidentally, this is a hot spot for genomic imprinting. So genomic imprinting is this pretty fascinating form of parent of origin expression that we see evolving in mammals and also has evolved independently in flowering plants. And this is where there's a shift from Mendelian expression, so expression of both alleles, to imprinted expression that follows the parent of origin pattern. Okay, so in general, this is mediated through various epigenetic mechanisms such as DNA promoter methylation or histone methylation or antisense transcription. But the really interesting thing is that it also shows this very non-random functional association. So paternally expressed genes, that is maternally imprinted genes, tend to promote growth, and then maternally expressed genes tend to repress growth. And so we think this actually is a partial solution to the inherent conflict. But this association has led people to propose very specific hypotheses related to how this mechanism may relate to these recurrent patterns of growth effects in mammals. And in particular, there's this leading loss of imprinting model. So the idea goes something like this. If you have loss of imprinting, then you would expect potentially dosage effects. So in this case here, where if we have loss of imprinting, we would activate this normally silenced allele, leading to an increase in overall expression. And then in a reciprocal cross, if you have a situation where imprinting is differentially lost from one of the two parental species, then you'll get parent of origin growth effects in the reciprocal cross. Now, this is a really interesting hypothesis, and it was, it's been around for a while, but it's really developed by Paul Verana and others working in deer mice, and they see some evidence for this. And it seems to be widely accepted in the literature, but if you actually look at the evidence for it, it's fairly variable. And there's some individual candidate genes that support this, but others that don't. And for example, recent work by Andy Clark and others has found no evidence of disrupted imprinting in crosses between horses and donkeys. And so we wanted to test this, and we wanted to do it in a novel model. And so we've been working in dwarf hamsters. So dwarf hamsters are a pretty well-established lab model for physiological experiments, and there's a lot of work on circadian and circannual rhythms in these guys. But there's not actually, they're undeveloped as genetic models. So this is work that's been led by Tom Brekke in my labs as a recently graduated PhD student. And we've been looking at these two closely related species of dwarf hamsters, Phodopus sangoris and Phodopus campbelli. And so these are very closely related species. They show a lot of really interesting ecological differences. Crosses between them produce hybrid male sterility. And if you dive into if you have somebody help you dive into the Russian literature, you can actually you can find evidence for growth effects, but they're not really well described. And so we first set out and did a series of crosses with these guys. And what we found, and indeed there was massive overgrowth effects in reciprocal crosses. So this is a plot of placental weight in these reciprocal crosses. So both parental species are on the outside here, and then in the middle we have the reciprocal cross. So in particular, when you have massive overgrowth when you cross a female sangoris to a male campbelli. So it results in about a 300% increase in placental size. This is a lethal cross, so it actually results in death of both offspring and mother in all the times that we've attempted it. And it's quite extreme, and also embryonic growth is quite extreme, but the placenta is particularly large. And when we look at the placenta, it actually histologically is fairly intact, and so there's not a lot of cellular differentiation between these different crosses. So kind of motivated by that, we've started to look at gene expression in these reciprocal crosses. And so I'm just going to kind of show you the patterns of differential expression. We compare placental expression between the species. We see a lot of differentially expressed genes between the species. But if we focus on those subset of genes that are just different between one of the hybrids and both of the parental species, so they're transgressive, they fall outside the range of the parental species. And we look at those, there's about 1,600 of those, and 1,400 of those are private to the large overgrown. So this asymmetric cross provides a really nice way to ask whether or not there's differential expression associated with the growth effects rather than just hybridization per se. And we have this massive effect of transgressive expression in this cross. Now, if we look at the kind of patterns of expression in more detail, we see that there's about equal numbers of genes that go up and down. And these are genes that make a lot of sense for these phenotypes, so it seems like the large hybrids are expressing a lot of things involved in immune response, probably inflammation, mitotic processes, that is a growth effect, and also a lot of things associated with tissue development. And so these are pretty strong effects, and there's also within this set, there's lots of known imprinted genes. So building on this, we've then used the fact that we have these RNA-seq data, and we can identify fixed differences between the species, and then we can use that to identify allele-specific patterns of expression. And so this is kind of a challenging field, and there's a lot of arguments about the best way to do this, so kind of using the best practices in the field. We could identify a conservative set of 88 autosomal imprinted genes by just identifying genes in the reciprocal crosses that show strong parent of origin bias, not species of origin, but parent of origin bias. Of these, 78% of those genes are differentially expressed, and so that's a massive enrichment for differential expression, so transgressively expressed. So vast majority of these imprinted genes appear to be really different in the hybrids. Of those, we then asked what subset of those showed changes in allele usage between the reciprocal hybrids, right? Because the prediction isn't just that imprinting is lost, and we would never see it anyway if it was just lost in both hybrids, but we want to actually identify things that appear imprinted in one hybrid, in particular the normal hybrid, and things that are biallelic or less skewed in the large hybrid. And there are 40 such genes. About half the genes show pretty big allele usage differences between the reciprocal hybrids. And this is consistent with disrupted imprinting. 39 of these show disrupted imprinting. 39 of these show disrupted paternal imprinting. So things that are normally expressed through the maternal line are biallelic, And of those, almost all of them are biallelic in the large. So this suggests that there's a connection between disrupted imprinting and the large growth phenotype. However, if we then look at the expression direction of these genes, we find actually that disrupted parental imprinting, so the gain of allelic expression, so shifting from bias towards just one parent towards biallelic, is associated with massively decreased expression for almost all of these genes. So I've plotted here the genes that are differentially, or that show these patterns of biallelic expression, and those above the line are higher expressed in the large, and those below the line are lower in the large. And the vast majority of these show really large log-fold changes in the large hybrids. And almost all of these are maternally expressed genes, so this is consistent with the disruption of paternal imprinting and massive reduction in expression. Now, the Dosage model for loss of imprinting predicts that this gain of allelic expression would be associated with increased expression. In the case for paternal imprinting, that you would actually expect a reduction in size, right? But this is not what we see. What we actually see is this gain of growth repressors is associated with dramatically reduced, almost universally reduced expression level and massive size effects. And so this actually is different than what you would predict for this loss of methylation. And we're actually working now, we're doing some methylation surveys and trying to get at this in more detail. And we think this actually might reflect a hypermethylation response. And so in the speciation literature has emphasized loss of methylation. But if you actually dive into the cancer literature, you see a lot of evidence for certain human diseases that are caused by normally imprinted genes becoming hypermethylated, and that leads to growth effects. So our data in particular are consistent, are functionally consistent with the growth effects, though, because we're having this reduction in genes that are generally associated with repressing growth, and the overall expression level of those are dropping, and that's consistent with the phenotypes that we see. So then we've been jumping into the genetic architecture. This is a non-model system, so we just kind of have to start from ground one. But we've been doing a bunch of crosses. We did introgression, mitochondrial introgression lines, where there's no effect of the mitochondria on size. We've done an initial back cross where we take F1 females and back cross them. So we mimic the large F1 hybrid, but we vary the maternal contribution. And so looking at 200 of these back cross offspring, we first do a QTL map, and what we see is actually, it's kind of nice for a change to have a simple genetic architecture for something. So we see just a massive X effect. And this is a parent of origin trait. That's perhaps not surprising. What I didn't tell you earlier is that X chromosome is imprinted, in rodents at least, and it's paternally imprinted. So all individuals express just the maternal X, and that imprinting is not disrupted. And so there's no evidence for disrupted X-linked imprinting. 2 minutes, right? So if you get the X chromosome from Sungorish, you're massively large. If you get the X chromosome from Cambly, you're small. So then what we've done is we've taken individuals with large placentas and normal placentas in this back cross, and we've repeated the RNA-seq experiment for a subset of these, for 24 of them. And we identify this core set of differentially expressed genes. And then we look at the overlap, and what we see is actually pretty remarkable overlap for this kind of experiment, with 165 genes that show the same patterns of expression in both genetic architectures. And these are highly enriched, and we actually identify all of the imprinted genes pretty much that we identified the first round. They show decreased expression in the large, and so this actually suggests that the X chromosome is interacting with the autosomes to lead to this disruption of paternal imprinting. So we think that there's this really interesting interaction. And this is pretty striking because actually in the house mouse and the deer mice where this has been looked at before, there is the same kind of large X effect. And so potentially there is a recurrent genetic architecture associated with disruption of placental function that's an interaction of the X chromosome with the imprinted autosomes. And just kind of to close out, so we're excited about this because we're actually beginning to hone in on another developmental mechanism that we think might underlie recurrent reproductive phenotypes in mammals, and we're excited that this actually might be related to this really fascinating process of genomic imprinting. And with that, I'll just thank Tom for all the great work he did on this, funding sources, and thank you.